First, we are told to gather at the bus stops before we start moving to Taman Botani Putrajaya and Taman Wetland. After a few minutes of the journey, we finally arrive at the Taman Wetland Putrajaya. After we arrived, we were given a few minutes of information about Taman Wetland Putrajaya. It's game time! The game is to find a few plants that were given in the booklet around the Taman Wetland Putrajaya. These are one of the plants that we successfully managed to search for. Next, we set foot on the Taman Botani Putrajaya to conduct Practical 1 and Practical 2, which is in the lab manual. This is bird's nest fern. The scientific name for this fern is Asplenium nidus. It often grows on other plants and is native to Southeast Asia, Australia and other tropical regions. Before we conduct our Practical 1 and Practical 2, we were given a tour around the Taman Botani Putrajaya, which is a very exciting experience for us. This is Javan rusa deer, also known as Sunda samba. The scientific name is Rusa timorensis. These deer are native to the islands of Java, Bali and Timor in Indonesia, and introduced populations can be found in various locations in the southern hemisphere. Now let's move back to the original objective, which is to conduct Practical 1 and Practical 2. Both are done in the same area.
Hello everyone, my name is Nusofana binti Mahdi and I will be explaining the finding from Practical 1. Let's go! So, what is quadrat? Quadrat is an ecological sampling unit consisting of a small square of ground within which all species of interest are noted or measurements taken. So, for this Practical 1, we have to take 10 leaves, measure and record their length and width. Then, calculate the mean, variance and standard deviation of the length and the width of the 10 leaves. Then, we have to repeat for another 2 sets of leaves, so we will have a 3 sets of leaves. And tada! This is the result of the set 1, set 2 and set 3. Next is post-lab questions. Question 1. Discuss the differences or similarity of the mean of length between different sets of leaves. Do the same for the mean of width. For the mean of length, set 1 has the highest average, which is 102.5 cm, followed by set 2 at 13.77 cm and set 3 at 12.72 cm. The average lengths of set 2 and set 3 are relatively close, differing by only 1.04 cm, suggesting a similarity in their size range, while set 1 stands out with a significantly larger value. Then, in terms of width, set 2 and set 3 also display similar average values, which is 4.32 cm and 4.1 cm respectively, whereas set 1 has a smallest width value at 3.83 cm. This indicates a more consistent width distribution between set 2 and set 3 compared to set 1. Overall, set 2 and set 3 share more similarities in both length and width, implying comparable structural characteristics or potential environment adaptations. Question 2. What is meant by variance? The leaves have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance. What is meant by standard deviation? Variance is a statistical term used to calculate how many different values of a data set are from the average value. The leaves have different measurement of width and length. The statement reflects biological type of leaf. Leaves are from different trees and different species will have different size and lengths depending on the need of the tree and how the tree will adapt to the environment. Taking measurement of the leaf from various types of tree from the same area will show us the spread out measurement from the mean. Low variance means that most leaves are almost the same size, while high variance means that the leaves have a large difference in size. Then, standard deviation is the square root of the variance. Variance gives a square difference of mean. Thus, standard deviation converts this information to an easier form of interpretation. High standard deviation means that the leaf sizes are different, while low standard deviation means that the sizes are almost similar. Question 3. Why is it necessary to have different sets of measurements for the measurement of leaf width and length? Is it necessary to have different set of measurement for leaf width and length because both dimensions reflect different aspects of how a plant interacts with its environment? This measurement helps to identify adaptation to factors such as light, water, and nutrient availability, giving a clearer understanding of plant function and survival strategies in different ecosystems.
Assalamualaikum. My name is Nafatin Fatiha and this is my Practical 2 report. What is population? Population is a group of organisms of the same species found within the same environment. How do scientists study the interaction of organisms? They do it by taking inventory of all species in the area and compare the sizes of their population. And this is the objectives of this practical. This is how quadrant looks like. Quadrant 1. Here is the placement of the trees from our observation and we use letters to differentiate them. For A, we have Hopia odorata. For B, we have Pomatin pinnata. C, we have Cytophilum fragrans. For D, we have Pentaspadon motley. For E, we have Neobaroneopus hemi. F, we have Paicia insignis. And G, Bambusa vulgaris. Here is the calculated density of each. We use the density formula, which is the number of trees divided by the total area of the quadrant. Quadrant 2. Here is the placement of trees that we observed. The name of the species are Roystonea regia for A. For B, we have Syzygium zelanicum. For C, we have Sinometra malacanis. D, Paricia insignis. E, we have Dirobalana sumatrensis. And F, Pomitia pinnata. Question 1. What is quadrat? And define the term in quadrat is a sampling tool that is regularly square or rectangular frame used to isolate and define area for the purpose of studying and quantifying the organism within the frame. What are the two basic requirements for all the quadrat technique? A, the pop of the quadrat must be determined exactly, and B, the area of the quadrat must be known. What is the advantages and disadvantages of using a larger or smaller quadrat? For large quadrat, the advantages is better representation of habitat variability, but the disadvantage is it takes more time and effort. For smaller quadrat, it is faster to sample, but the result will be biased. The ecologists are having a problem in conducting the sampling method. What are the issues? They could be 1. Limited visibility and accessibility. 2. Scale mismatch. And 3. Sampling bias. Why is there a difference in the number of plant species? And discuss the factors. The hypothesis that we could make is the abiotic factors, which is environmental factors such as sunlight, availability, soil moisture, nutrients level, and human disturbance. First is sunlight. Some quadrats may receive more sunlight due to their position or lack of canopy cover allowing sun-loving species to thrive, while shaded areas may support only shade-tolerant species. Second is availability. The presence or absence of essential resources like water, nutrients, or space can influence which plant species are able to establish and thrive. Limited availability may restrict growth, while high availability can support a wider variety of plants. Fourth is soil moisture. Areas with higher soil moisture can support a different range of plant species compared to drier areas. Plants that need more water will only grow where moisture is sufficient. Last but not least is human disturbance. Activities like trampling, clearing, or pollution can alter plant diversity. Disturbed areas might show fewer species or a dominance of fast-growing, resilient plants.